Back to my biochemistry video. Today we're going to be going over antibodies, and I'd like to produce some antibodies of my own to attach to biochemistry and neutralize that subject. However, we don't always get what we wish for. So, moving on, what exactly is an antibody? Well, an antibody consists of polypeptide chains connected via disulfide bonds. And there are two types of antibodies we really need to look at, polyclonal and monoclonal. Now, how do antibodies actually work? They bind specifically to various epitopes present on antigens. So antigens have a portion of them called an epitope, and that's where the antibody binds. Now, antibodies are very specific to what they bind to. They can only bind to one epitope. They cannot bind to various epitopes. One antibody per one epitope. That's how to think of it. So the difference between monoclonal and polyclonal is monoclonal antibodies come from the same lineage, so they are basically identical twins. Now, monoclonal antibodies bind to the exact same epitope. Polyclonal antibodies are basically friends, or they're distant relatives. They do not bind to the same epitope. They bind to different epitopes. So monoclonal binds to the same epitope. Polyclonal, they bind to various epitopes. But don't think of it as each antibody has the ability to bind to various epitopes if it's polyclonal. Polyclonal is basically just a mixture of antibodies with each binding to its own unique epitope. Monoclonal is basically clonal lineage. So yeah, think of Star Wars: The Clone Wars for monoclonal, and polyclonal, think of just a bunch of random people at a party. Once again, we don't really care about polyclonal antibodies because they're not specific enough for us to use. It's basically just a bunch of random antibodies thrown at us. Monoclonal, once again, all attached to the same epitope, so they are actually highly desired in the biotechnology field. And pharmaceutical companies love monoclonal antibodies because they can make a lot of money off monoclonal antibodies, because monoclonal antibodies can be utilized to treat infections and help people that are actively infected by boosting their immune response. So, since monoclonal antibodies are so desired, how do we actually isolate them and grow them in a lab? So the first thing you're going to want to do is basically infect a rat. You infect a rat with, say, pathogens such as HIV. Once the rat is infected with HIV, we can allow that rat to sit for about 4 days and let his adaptive immune response to kick in. Once his adaptive immune response kick in, there's going to be an abundance of lymphocytes, B cells, which produce antibodies, specifically plasma cells, which produce the antibodies, present in his spleen. So we can then harvest his spleen, or harvest his spleen cells. We're not actually going to cut out Mickey Mouse's spleen, don't worry, kids. We can harvest his spleen cells, then we can mix those with myeloma cells. Now, if you guys understand how cancer works, myeloma cells is a type of cancerous cells, and cancerous cells rapidly proliferate. That's actually what causes cancer. So once we mix these spleen cells with the myeloma cells, you'll form what's called a hybridoma cell. Now, hybridoma cells constantly secrete the antibody. They're basically cancerous antibody-producing cells. And since antibodies rapidly proliferate, we have rapidly proliferating cells producing the antibody in question we might want. We don't know if we want it yet. We haven't tested it. But hybridoma cells are rapidly proliferating antibody-secreting cells. Once we have those cells, we can actually screen for the desired hybridoma cells utilizing the methods I'll talk about here in the next couple slides. But once we actually isolate the desired hybridoma cell producing the antibody we want, we can then either induce a tumor in Mickey Mouse and then harvest the antibodies from that tumor, because tumors are rapidly proliferating cells. And if we induce a tumor via hybridoma cells, we'd have rapidly proliferating hybridoma cells. Or we can grow them in mass cultures and such, like Petri discs and stuff like that, kind of like Henrietta cells. But yeah, that's how you isolate a monoclonal antibody. There are actually 3 ways to detect an antibody. It doesn't have to be a monoclonal antibody. It could be polyclonal antibodies also. But there are 3 ways to detect an antibody. There's direct ELISA, indirect ELISA, and Western blotting. These are the 3 main methods to use. Indirect ELISA this is also called sandwich ELISA. Direct ELISA is called sandwich ELISA. Should note that. Indirect ELISA is the most efficient, and then direct ELISA is the least efficient. So taking a closer look at indirect ELISA. So indirect ELISA actually works by having a beaker with antigens lined at the bottom of it. Now, as we know, antibodies are antigen specific. They only bind to one type of antigen. So if we know what infection we are dealing with, and we want the antibodies that helps neutralize that infection, we can produce the antigen that causes that infection at the bottom of this beaker. Now, the antibodies we want in question to neutralize this antibody should, in theory, attach to that antigen, because once again, antibodies only attach to specific antigens. So how we do this is we will put a bunch of antibodies in this beaker, and the antibodies we want will bind to the antigen at the bottom of the beaker. If none of these antigens bind, or antibodies bind to the antigen, then our antibody was not in the solution we isolated, and we know we failed. However, to test whether our antibody is actually present in the solution, we can add antibodies that are laced with a specific enzyme. Now, these antibodies that are laced with a specific enzyme will bind to the antibody that actually bound to our antigen. Once this occurs, a change in coloration happens, and if we actually can visualize this change in coloration, we know that our antibody is present in the solution, and then we can go on to isolate it. So the main concept to know from this is the indirect ELISA is utilized to detect whether our desired antibody is present by having the bottom of a beaker laced with antigens and having our desired antibody have the specific ability to bind to that antigen, and then we can actually know whether that antibody is present by adding enzyme-laced antibodies that attach to our desired antibody, which then cause a change in coloration. So sandwich ELISA is called sandwich ELISA because it kind of looks like a sandwich, as you can see. And how this works is it usually is utilized in order to detect antigen quantity. So if we're trying to isolate an antigen in question, we will usually utilize sandwich ELISA. However, you can use it to determine whether your antibody is present. You would just have to put the antibodies at the bottom of the jar that you want, and then you would have to expose them to the antigens that you know that antibody can neutralize. However, that's much less efficient than utilizing the indirect ELISA. So, how does this actually work? Like I said, you have antibodies at the bottom of the jar that you know attach to specific antigens, because once again, antibodies are antigen specific. The antigens will then bind to the antibody. When these antigens bind to the antibody, we can then add the antibodies with the enzyme attached to them to the solution. These enzyme attached antibodies will then also bind to the antibody, forming a solution sandwich-like structure. And if the coloration changes, that means that the antibody that we want and the antigen that we want is actually present due to that, once again, that color change. So the easiest way to compare the two is sandwich ELISA is utilized in order to detect antigens. Indirect ELISA is utilized in order to detect the antibody. However, both can be utilized for either. It's just that the main concept is like what I just previously said. Final thing we're going to be talking about is Western blot. Now, Western blot actually utilizes SDS gel electrophoresis, and if you remember from my previous video what that is, that will be kind of helpful, but once again, you don't even really have to understand it for this concept. Western blot is utilized in order to detect small quantities of protein in cellular fluid or human fluids. And how this actually works is you start with your SDS polysaccharide plate, your SDS gel electrophoresis plate, and you transfer the proteins to a polymer sheet. Once they're on a polymer sheet, you can actually submerge the polymer sheet with these proteins into an antibody solution. So in this solution right here, there's a bunch of random antibodies floating around. And as we know, the proteins on here would be antigens. So then we can have the antibodies attached to the antigens if it is the desired antibody, because once again, that specificity. And then we can actually take out this polymer sheet that now has the antibodies attached to the antigens, and we can utilize spectroscopy or mass spectrometry in order to detect whether that antibody or antigen is present on that plate, because the spectroscopy reading would be a specific reading in order for us to determine it. And yeah, that is basically Western blot. The main concept to know is just basically the definition of Western blot, which is it is a type of test utilized in order to seclude small amounts of proteins from human fluids and cells fluids. And it utilizes SDS page, gel electrophoresis, and it utilizes a polymer sheet. And then that polymer sheet has the antigens present on it. And then when you put that polymer sheet with the antigens present on it and a solution of antibodies, the antibodies will bind to the antigens if they are able to. And then we can actually then detect if our desired antibody bound to the antigen by undergoing spectroscopy. And yeah, that's basically all there is to antibodies, guys. I hope this video was helpful and you guys have a wonderful day.